Dear students, welcome to the online classes of biotechnology. Today I am going to explain you about expression vectors and the production of recombinant proteins that is called R proteins, recombinant proteins. So first you should know the difference between cloning vector and expression vector. So far I have explained you about several vectors. Few of them were cloning vector and few of them were expression vectors. So if you see the definition of cloning vector, a cloning vector is a small piece of DNA which is used to introduce foreign gene of interest. So it is only used to introduce the gene of interest in the host. However, an expression vector is a plasmid that not only introduce gene of interest into host organism but also produce the relevant protein or other products in the host system. If you talk about the role, the cloning vector is used to obtain multiple copies of the foreign gene of interest in the host. However, the expression vector is used to obtain the final gene product like protein. So if you want to express the protein, you should clone your gene of interest in expression vector only. If you see the types of cloning vector, I have discussed various cosmid vectors, phasage, and yeast artificial chromosomes. But the expression vector can be always like plasmid vector. If you see the feature of cloning vector, I already discussed in previous lectures that a typical cloning vector consists of origin of replication, that is ORIC, unique restriction sites, reporter gene, and antibiotic resistance site. Expression vector also contain these special features. Apart from that, they should contain some additional features like it should contain promoter, enhancer, terminator, and transcription initiation sites. So these additional features make a plasmid vector or cloning vector it can convert into the expression vector. So here, as I already discussed, there are three most important signals, three main important features for the expression vector. The first one is promoter. Actually, promoter is the point at which transcription of genes should start. So in E. coli, the promoter is recognized by G subunit of transcribing enzyme RNA polymerase. Here I have shown you the promoter region here. So your gene of interest should be always cloned between the promoter region and the terminator region. Here you can see this gene of interest is cloned between this promoter region and terminator region. Second is terminator region. Actually, it is the point at the end of the gene where transcription should stop. So this is also important because if you will not put terminator region, so your gene will continuously transcribe, or your gene of interest or your final product, that is protein, will be much longer than you expect. So the terminator region is also important. And third important thing is RBS, that is ribosome binding site. It is a short nucleotide sequence recognized by ribosome as the point at which it should attach to the mRNA. So during the protein synthesis or translation, the first ribosome actually will bind in the RBS region. So here I have shown you some example of promoter region. Actually, the promoter region of E. coli start from -35 to -10. Minus means if your gene of interest is here, means first signal sequence, that is initiation codon ATG is here. From this 10 nucleotide upward, means up to the 5' region, up to 35 nucleotide here. So this is called promoter region from -35 to -10. So in -10 region there is a data box in the case of E. coli. So similarly here, the promoter of various animals, you can see the data box is present in -25 region. So that's about the promoter and terminator regions. And you can also convert any plasmid into the expression vector. Here I have shown you how you can make any plasmid in, and you can convert into the expression vector. Here you can see this is the P stands for promoter. You can introduce any promoter, like I have shown you various example of promoters here, like leg promoter, trap promoter, tag promoter, T7 promoter. So you can introduce this T7 promoter in the plasmid, and you can also introduce the unique restriction site here, and finally you can introduce terminator site. So if you digest it with unique restriction enzyme, and you can ligate the gene of interest here, and you can transform this clone plasmid, this gene with this cloned gene, and transform into the E. coli. What happens in E. coli? Actually this plasmid actually contain the promoter, and downstream of this promoter region you have cloned your gene of interest, and in E. coli this gene will actually express in the form of mRNA, and finally it will form the protein in the E. coli. Suppose you have cloned the human gene here, so your human gene or insulin, any human gene like insulin, will produce insulin inside the bacterial system. So that's the beauty of this expression vector. You can clone and express any foreign protein or any foreign DNA into the E. coli or heterogeneous system. Like here you can see the gene of interest, like human gene, is expressed in the E. coli. So the most important promoter is actually T7 promoter and leg promoter. So I am going to explain them separately here. So the leg promoter, actually I discussed this already when I discussed about the puck vector. So the leg promoter is present in the puck vector, and it is induced by the IPTG, and it is a strong promoter as well. And I have discussed about this leg gene when I explained the puck vectors. Second important promoter is T7 promoter, and this promoter is actually very strong promoter, means it can efficiently transcribe into host cell. And T7 is actually the DNA region from the T7 phage. When we introduce a T7 DNA into the plasmid, it can convert into the T7 promoter containing vector, and it is very efficiently transcribed vector. So T7 promoter is generally used for the expression of various heterogeneous gene or foreign gene into the E. coli. Now I have shown you an example of expression vector here. This is the PET vector. We have also this vector in our laboratory in the Department of Zoology. And here I have shown you how we can clone a gene of interest or foreign gene into this bacterial expression system. So here you can see this is a T7 promoter containing vector. So this is T7 promoter, and there is a gene, and this gene is actually any gene. I have shown you an example of a PPG gene that I have cloned during my PhD time. So here you can see how this gene can be cloned in two ways, like wrong orientation and right orientation. So when it is cloned in wrong orientation means the terminator codon, this is TAA, is near to your promoter region. So what happened when the gene will be cloned in this orientation, that is wrong orientation, it will not produce the protein because it will terminate before it starts the transcription. And on the other hand, you can see when the gene of interest is cloned in right orientation, you can see here the start codon is near to the promoter region, so your gene will express continuously, and here it will terminate at the termination codon. So how you can check your gene of interest is cloned in right direction or in wrong orientation? So for that you have to search for an restriction site, actually here, that is XHO1, which can digest this gene of interest at suppose this is the gene of 1593 base pair. So suppose XHO1 is digesting at 1074 base apart from your start codon here. From here to here, this is 1074, and suppose this XHO1 is also digesting the vector. So when you digest this plasmid, when you transform the cloned plasmid in the E. coli, and it will grow in the selection marker, and you can isolate the plasmid and you can digest the plasmid. So there are two possibilities. One is wrong oriented plasmid, another is right orientation plasmid. So in wrong orientation, when you digest with XHO1, you can get two bands, actually one band of this 11-14 base pair, and rest remaining is 5.8 base pair, because the size of this PET vector is 5.3 kilo base pair. So in wrong orientation you will get two bands, that will be 5.8 kb and 1.1 kb. However, in the case of right orientation, you will get two bands, one is 6.4 and one other is XHO1 digested fragment, that is only 5.5 kilo base pair. So you can practice this at your home, and this is quite important for your CSIR net examination also. Always they can ask the question about this, how you can say the gene is cloned in right orientation and wrong orientation. So for this you have to practice at your home in your notebook. So if you have a query, you can also ask in Google Classroom, or you can even call me regarding this right orientation and wrong orientation problems. So here now I am showing how this gene of 1593 base pair, it can produce the protein, and the size of that protein will be 58 kilodalton. Why? Because one amino acid, the molecular weight of one amino acid is 110 dalton, and one amino acid coded by three bases, like ATG is coded for methionine, can code for methionine. So 1593, I have given you example of this gene which is 1593 base, so it can code for 531 amino acids, and average molecular weight of that protein will be 531, that is the number of amino acids, multiplied by 110, so that will be 58,410 dalton. It will equivalent to 58.41 kilodalton. So the gene of 1593 will produce a protein that will be 58 kilodalton. And finally, I have shown you some important enzymes which are used in the molecular biology lab. So as you know, first one is DNA polymerase, DNA pol 1, clean-off fragment, T4 DNA polymerase, tech DNA polymerase. These are the very important DNA polymerases which we use in the molecular biology laboratory for cloning and expression purpose. Apart from that, there are some RNA polymerases, nucleases. Nucleases are like exonucleases, endonucleases. I have given you several examples of restriction endonucleases as well. Apart from that, reverse transcriptase, transferases, ligases like T4 DNA ligase, which we use for the ligating the gene of interest into the vector. Similarly, kinases is also important, phosphatases and transferases. So these are the list of some important enzymes for molecular biology experiments. So you can read it properly. So that's all about today's lecture. If you have any doubt, you can ask me on my WhatsApp number. Till then, next time, actually, I will start a new topic. I think I will start a PCR. So, till then, stay home, stay safe. Thank you.